Welcome, dear students. Today in chapter 4, we need to learn more about the different mechanisms of the antibodies, and starting with the clumping and how they are used to stop the action of the pathogen. Let's start our lesson today. Acquired or adaptive immunity. In our lesson today, we need to know what will happen if the first line of defense failed in protecting our body. If the second line of defense failed in getting rid of the foreign body or to remove and destroy the pathogen, the body will use the third line of defense, which including what? The lymphocytes. These defense mechanisms are collectively, we call them the immune response. But why they are called this type of immunity immune response? This acquired or specific immunity is done by two different mechanisms, which they are: the first mechanism, humoral or antibody, and we call it mediated immunity. And these mechanisms defend the body against antigens such as what? Like bacteria, like viruses, and like toxins. I mean the different types of pathogens. And this type is found in the body fluids such as blood plasma and lymph. They are active by producing the antibodies. And this is done on the following steps. When the pathogen enters the body carrying on its surface, as we started in the last episode, a specific antigen, the B lymphocytes recognize this antigen, or definitely the B lymphocytes collect information and gathering some details about this pathogen, about the antigen that they are produced by the pathogen. Each B lymphocyte is very specific and can also respond to a single specific antigen. But what happens when the B lymphocyte recognizes and collects information to its specific antigen? It attaches itself to the antigen. So number one is attachment, by using what? By using the immune response. And also the CD19 and CD20 or CD21, that they are referred to what? Those clusters of differentiation, they are referred to, which is what? The clusters of differentiation. And also they have important role by helping of B lymphocytes. After that, after attachment and the clusters of differentiation, and they pass them to the lysosomal enzymes into fragments or very tiny pieces. These fragments bind inside the macrophages to protein. This protein is called MHC2 or histocompatibility complex. The complex resulting from the binding between the antigen and the MHC2 or major histocompatibility complex to transfer it to the plasma membrane of the macrophage to be presented on its outer surface to the T helper lymphocyte. After that, the T helper lymphocytes will recognize the antigen by the MHC protein that present on the surface of the macrophages and bind to the antigen, which is the major histocompatibility, which this complex it directly bind This binding activates the helper T cells to release the interleukins, okay? And the interleukins will activate or the response, the B cells carrying on its surface the antigens bound to the what? The major histocompatibility complex. And after that, this is a picture to show us what the antigen that present in the macrophage, and it displays the antigen fragments are stopping its action on the surface of the receptors, and also the macrophage where it's engulfing the antigen. After that, what happened to the T helper? Talking about the function of the T helper in this process and how they are recognize the antigen by the MHC protein and also by binding with the MHC2 complex. The helper T cells can recognize the antigen only after being treated by the macrophage. So the helper T cells at the first they cannot bind with the antigen, but after they treated by the macrophages they are very reliable and they are presenting it on its outer surface and they are bound to the major histocompatibility complex. So the function of the active B cells at this moment is what? To activate the B cells with divide and multiply and differentiate into a memory cells. And remember that the memory cells, they have a very function role in this process and also in the plasma cells. So again, the multiplying or differentiation into memory cells and plasma cells, which produce after that a large amount of antibodies. These antibodies pass through lymph vessels and the blood circulation to what? To fight and defend the infection. The memory cells will stay or remain in the blood for a very long time, for a time of period about from 20 to 30 years, for doing what? To recognize the same antigen if it re-entered the body. So if I'm going to ask you the function of the memory cells, they're doing what? They recognize the same antigen if it re-entered your body again. They also divide and differentiate into plasma cells that secrete antibodies that's specific for the same antigen after a long time, and making a rapid response, that faster than the first response to the first infection if the same antigen is re-entered again, okay? The healing of this process will be more faster than before. After that, we need to know the role of the antibodies. What is the role of the antibodies? Well, actually, the antibody, they have a lot of role inside our body, such as what? They produce the antibody produced by the plasma cells will reach the blood circulation through the lymph, and also the binding to the antigen that they are found on the surface of the invading pathogens, and they also bind to the antigens they are found on the surface of the invading pathogens. So this will doing what? This will activate the macrophages to what? To re-engulf these antigens again more than one time. Yet this will continue for days or weeks. Okay, and we have here a picture just to show us what happened, or just the rule to show us the rule of the antibodies. Okay, we have here what? We have different types of the antibodies, and each one of them has its own role of what? Of engulfing and destroying the pathogens and their antigens inside your body. And after that, we have a question here for you. If I asked you the antibodies produced, why the antibody produced by plasma cells aren't effective enough to destroy foreign cells, such as the cell they are infected by viruses. Again, the question here, why the antibodies produced by the plasma membrane, they aren't effective enough to destroy foreign cells like infected by the viruses. Okay, and to know the answer on this question, you have to know that the antibodies are relatively large in size. They are very large in size, okay? I mean, relative to another molecules. They are large in size, but molecules that cannot reach the virus inside the cell. So their larger size make them a very difficult, very hard to penetrate the size of the cell. In this case, the foreign bodies will be competed by the T lymphocytes. Well, cellular or cell-mediated immunity. Here is it, the second type of the cellular or the cell-mediated immunity, which includes what? The immune response done by T lymphocytes, and also which is done by the receptors that are found on its membranes that give it a specific response to what? To the antigen. And also that each T cell, they have ability or during its maturation and a specific type of receptors to specific to its membranes. And also each T cells, they can produce during its maturation, I mean when they are grow more and more, and a specific type of receptors for a specific to its membranes. Each of these receptors can bind to a single type of antigen, okay, just a single type of antigen that represented by what? The macrophages. These mechanisms can be summarized as the following. Okay, we have here just a summary for what? For this mechanism by more than one step. Well, step number one. When the pathogen or bacteria, like bacteria or virus, whatever, they enter the body, what will happen? Number one, the macrophages will engulf it and decompose it into small fragments, small pieces. These small pieces that bind inside the macrophages to form what? To form major histocompatibility complex. Yet, the antigen, okay, and major histocompatibility complex will transfer to the plasma membrane of the cell to be presented on its outer surface. While the T helper lymphocytes, they have the receptors, which is CD4, I mean the cluster differentiation, and its membrane will bind to this antigen and the major histocompatibility complex that present on the surface of the macrophage. But what happens when its CD4 or cluster differentiation receptor binds to this complex? There is something should be happening here. Actually, this binding will activate the helper T cells, and also the activation of T helper cells will release the interleukins to activate the Th cells. So the interleukins bound to the antigen to divide to form strands of activated helper T cells and also memory cells that last in the blood for a long time. These memory cells spend long time they are staying in the blood to recognize the previous antigen if it entered, as we said it before, if it re-entered in the body again, just to recognize it, I mean to collect information about it. And we have here a picture to show us the mechanism of invasion of bacteria or pathogen and how the membrane cells remain inside the It remains inside the blood for a longer time after the infection. Well, after that, we need to know the function of activated T helper cells. The function of activated T helper cells. Well, number one, they secrete different types of the proteins, like cytokines that do some functions inside our body, like what? Like attraction. They attract the macrophages to the site of infection in the large amounts. So they have main role in what? In attracting the macrophages. They call the macrophage to the site of infection, to the site of presence of the pathogens with a large amount. Also, they stimulate, they activate the macrophages and other types of the T lymphocytes, and like B lymphocytes. So they're activating both of the cellular and humoral immunity. Again, one of its most important function is stimulation or activation different types of T lymphocytes and B lymphocytes. So they activate both the cellular and humoral immunity. Also, they activate the natural killer cells to attack the abnormal body cells like cancer cells or cells that maybe some cells that infect us like pathogens. And also to recognize the antigens bound to the major histocompatibility complex. And also to activate the cytotoxic, which is T cells. All of that, they are the function of the activated T helper cells. Well, after that, the function of the cytotoxic T cells. Well, their function is restricted in two ways. They can recognize foreign bodies by the help of the receptors cluster differentiation 8 that found in its surface. And also, these foreign bodies are transplanted tissues or the antigens of the microbes that enters the body or cancer cells, and they can destroy them from the body. We have here a point about that. What happens when these cells bind to the antigen? There is something will be happen. Yes. Number one, they create pores in the membrane of the foreign body, or like microbes or cancer cells, whatever. By secreting these pores, they secrete a specific proteins, okay, like what? Pre-fermenting proteins. And this protein, or also they can secrete lymphatic toxins that activate these toxics, or these lymphatic toxics. They have ability to activate a certain genes in the nucleus of the infected cells. That causes what? That leading to destruction and destroying of the nucleus and its death. Finally, we have here, if I'm going to ask about what happened after destroying the antigen, we need to understand what will happen after destroying the antigen. I mean, if it stay inside your body or destroy or removed or eliminates, what happened after destroying the antigens? The T suppressor cells bind with the help of the receptors cluster differentiation 8 found in its surface to the plasma cells. The T helper cells and T cytotoxic cells, both of them, this binding will help to secrete a protein, this protein called lymphocytes. The plasma cells will stop in producing antibodies. Many of the T helper cells and the activated T cytotoxic cells will die. After that, some of them will be stored inside the lymphatic organs where they stay ready to combat or to kill, to attack any similar infection when they need it. And we have here a picture to clarify what did we say right now after destroying the antigen, okay, and have the lymphokines suppressor or inhibit the immune response or stop it, and also the many of the T helper cells that activate the T cytotoxic cells and destroy them. Well, we have here the stages of the acquired immunity. What are the stages of acquired immunity? Actually, we have two stages. First and second, one of them is the primary immune response, and the second one, which is the secondary immune response. Let's start it. The acquired immunity, it is the resistance of the body to pathogens that infected the body before, and it passes through two stages. The first one, we have a give reason here just to remind you. When somebody is infected with a specific disease like measles, high fever, he will not be infected again by the same disease along his lifetime. Well, because he gains acquired immunity against the diseases. Do you remember the memory cells? It is the resistance of the body to pathogens that infected the body before, and it also passes through two stages. The first stage, so we have someone he infected by a pathogen or something like that, and after that, if after a long time when he exposed to the same pathogen, he will never infect again. Well, let's to know it. Number one, the first stage or the primary immune response. When the immune system encounters a new pathogen, okay, so we have something new right now. We didn't infect it before. What happened? The B and T cells will respond to the antigens of this pathogen, this new pathogen, okay, that the body it doesn't recognize it yet. After that, attack it, and it's going to attack it until it is what? Destroyed. So this takes a longer time, since these cells need time to what? To multiply and to divide more than one time for attacking and engulfing or destroying this pathogen. The first response takes between 5 to 10 days. It's too much to reach its maximum productivity of B and Y cells. So we don't need to take much more time. But during this time, the infection could be widespread and the symptoms of the diseases will be appear. And this is a diagram that to show the first infection and the second infection. Okay, the first infection, it takes much more time, okay, because it needs a longer time for multiplication of the required cells or B lymphocytes and so on. While in the second immune response, let's to know what happened in the second infection or what will happen in the second stage. Well, the second stage or the secondary immune response, if the same individual is infected by the same diseases again, what will happen? So we need here to summarize, to lessen the time of infection and at the same time the time of healing. Well, the immune response will be very fast, but for what? That the pathogen is destroyed before the appearance of the symptoms. That what happened in the second stage. Let's to know it. The cells responsible for the secondary immune response are known as what? The memory cells. Do you remember that? They stay in the blood or in your body for a longer time, from 20 years to 30 years, and maybe sometime more. These memory cells store some information about the antigens to what? To fight and also to fought by the immune system, okay, to recognize by the immune system in the old time or in the past. Your body contains both memory, okay, B cells and memory T cells. So we have here some information they already collected before by the memory cells, and they are archived inside your body by a specific type of the cells. And after the infection, both types of memory cells are produced during the primary immune response, and B and T cells can survive only for few days. But memory cells can live for tens of years, as we said it before, from 20 to 30 years and maybe more, and also may survive till your death. But what happened during the second infection with the same pathogen? There is something happening here. The memory cells respond to the pathogen once it enters the body. Again, the memory cells, as soon as the pathogen enters the body, they are activated and start dividing quickly more than one time to produce a large amount of the antibodies for these pathogens, okay? And the active T cells within short period of time, okay, as we said, the both types of memory cells are produced during the primary immune response, okay? And after that, the B and T cells can survive for a few days. Remember that. We have some cells, they can survive for a long time, like what? Like memory cells, as we said, they can spend more than from 20 to 30 years inside your body, and sometimes they may remain till the death, okay? They can continue like your life. And after that, memory cells can live for tens of years, okay? So we have here three types. B and T, they can just survive for what? For several days, while memory cells, they live for a long time. But what happened during the second infection with the same pathogen? The memory cells, when they respond to the pathogens, okay, I mean, as soon as the pathogens which you are already have been infected by it before, okay, I mean, it's an old history of your disease, the memory cells directly deactivated, and as soon as the pathogen enters the body, they're doing what? They starting in dividing quickly more than one time to produce a large amount of the antibodies. But remember that the antibodies they are produced, they are already gathering of information. They already the information about these antibodies they already found in the memory cells before. Okay, if the pathogen is for the first time you're infected by it, the memory cells cannot gather and collect information, and they cannot divide more than one time quickly to produce antibody suitable for this antigen or this pathogen. But it should be necessary, okay, to be what? For the memory cells to be you already had been infected by this pathogen or this infection before. And after division, the active T cells within a short period of time. Within what? A short period of time, not more than that. Okay, and let me to remind you that we have two stages here, from the first infection and the second infection. As we said, the first infection, it takes much more time, long time, while in case of the second infection that we have memory cells, they are found inside your body, the collecting information, and when the pathogens that you already had been entered before or had been infected by it before, they divide more than one time and quickly. They divide, the multiplication will be faster more than before, and finally the infection of it and the destroying and destruction of this pathogen will be faster than the first time of the infection.